Okay, today we are going to TRCRC in Elmina for our field trip for BIO464 process in ecology. Now I will show to you about the video that we measure about the trees and anything. After that, the guide take us to the pot that they collect the seed from the forest to take the seed and make them grow in that pot. We have got so many information from the guidance and so many that we learn about the trees from that day.
Okay, for the objective for this practical one is to calculate and describe the mean, variance and standard deviation of biological measurement. Second is to understand and concept of correlation between two variables. And third one is to determine the type of relationship between leaf length and width, whether linear or otherwise. After that is method. First thing is collect 10 leaf, measure and record the length and width. Calculate the mean, variance and standard deviation, which is SD, of the length and the width of the 10 leaf. After that, tabulate data in a graph. After that is repeat for another 2 sets of leaf, so each will have 3 sets, which gives us 30 leaf from the same tree. After that is use data of all 30 leaf to plot a graph of leaf length versus leaf width. Okay. Tree code is S705 T190. Height is 99 centimeter. And width 55 centimeter. For root is 14 millimeter. The second one is tree code S350 T020. For height is 142 centimeter. Width 34 centimeter. And root is 10 millimeter. Okay, for the first thing is set 1, leaf length. For the mean is 11.1 cm and the variance is 7.9. Standard deviation is 2.81. After that is set 1b, which is leaf width. For the mean is 4.51 and variance is 0.4543 and the standard deviation is 0.6740. After that is set 2, leaf length, which is A. Mean is 11.24 cm and the variance is 6.20 and the standard deviation is 2.49. After that is B in set 2, which is leaf width. For the mean is 4.49 and after that is variance is 0.3108 and the standard deviation is 0.5575. And for the last is set 3. For part A is leaf length. For the mean is 12.05, which is the highest in mean. After that is variance 3.3628 and after that is standard deviation, which is 1.8338. After that is B in set 3, which is leaf width. For mean is 4.18 and for the variance is 0.484 and for the standard deviation is 0.6957. After that is post lab in practical 1. Question 1 is discuss the difference of similarity of the mean of length between different set of leaf. Do the same for the mean of width. We got the mean leaf length for set 1 is 11.1 cm, for set 2 is 11.24 cm, and for set 3 is 12.05 cm. There is a clear difference in the average leaf length across the 3 sets. Set 3 has the longest leaf on average, while set 1 has the shortest. This difference may be due to the leaf morphology. Mean leaf width is for set 1 is 4.51 cm, and for set 2 is 4.49 cm, and for set 3 is 4.18 cm. Set 1 has the widest leaf, while set 3 has the smallest width. This indicates that leaves from set 1 are generally larger width compared to the others. For question 2, in practical 1 is what is mean by variance. The leaf have different measurement of width and length. What is the meaning of this statement? Discuss with respect to variance what is mean by standard deviation. For me, variation is the average of the squared difference from mean. It measures the spread or dispersion of the data. A high variance means the data points are more spread out from the mean, while a low variance means they are closer to the mean. The statement, the leaf have different measurement of width and length, means that there is natural variation in the size of the leaf, even though they are from the same tree. This variation is quantified by the variance value. For example, set 1 length variance is 7.99000. For set 2 length variance is 6.2000, and for set 3 length variance is 3.3628. Set 1 shows the highest variance, indicating greater variation in leaf length. Standard deviation SD is the square root of the variance. It also measures how much the value deviate from the mean, but the same units are as the data. This makes it easier to interpret compared to variance. For question 3, in the practical one, what is it necessary to have different sets of measurement for the measurement of leaf width and length? For me, having a multiple set of measurement is important because it improves the accuracy and reliability of the result by giving better overall picture of the natural variation in leaf size. It helps identify consistent pattern in the data and reduce the influence by any outliers or unusual values that may affect the result if only one set is used. For practical 2, objective is to determine the plant species diversity and density at different sites in the Elmina Rainforest Knowledge Center, which is located in Elmina, using quadrat sampling method. For method, we use quadrat size 5 meter times 5 meter. Plant in quadrat were identified and count. Observe and take note all three stand and are found inside your quadrat. We have collected the data, which is number 1 is Saraka indica, which is in the bone. Total number of individuals is 1. Density is 0.0167. Number 2 is Ficus racemosa, which is in symbol banana. Total number of individual is 1. Density is 0.167. The third one is Pometia pinnata, which is in the cat. Total number is a 1 and the density is 0.0167. The fourth one is Delenia indica, which is in the button. Total number is 2 and the density is 0.0333. After that is fifth, which is Pometia pinnata in the symbol of apple. The number is 1 and the density is 0.0167. The sixth one is Meletia hamsliana, which is in the leaf. The total number is 2 and the density is 0.0333. And the seventh is Sia palembanica, which is in the dinosaur. Total number is 1 and the density is 0.0167. And for the eighth one is Aquilaria malasensis, which is in the bear. Total number is 1 and the density is 0.0167. And the next one is Terminalia calamansani, which is in the bread. Total number is 1 and the density is 0.0167. After that, for the last one is Artocarpus integer, which is in the broken heart. Total number is 1 and the density is 0.0167. After that is post-like question for practical 2. For the first question, define the term quadrat in practice. A quadrat is a square-shaped tool with a fixed area, commonly 5 meter times 5 meter, used in ecology survey. It helps researchers observe and record the types and number of plant, species or non-moving organisms in a certain area. In practice, quadrats are used to estimate population size, species presence and overall biodiversity in a habitat. For question 2, what are the two basic requirements of all quadrat technique? Quadrats should place randomly or in a clear pattern to make sure the data is fair and not biased. The size and shape of each quadrat must be the same, so the results are consistent and easy to compare. After that is number 3 question. What would be the advantage and disadvantage of using a larger or smaller quadrat to survey the sedentary organism? For larger quadrat, advantage is can record more species, which give a better picture of biodiversity. For disadvantage, takes more time to observe and can be harder to manage. For smaller quadrat, the advantage is quicker to use and easier to handle in the field. For disadvantage is may miss some species, so the result might not represent the whole area well. After that is question number 4. What is the problem with spatial scale in habitat like forest or aquatic environment? Ecologists often face challenge when studying larger or complex habitat like forest or water area. Access can be difficult because thick plant, uneven ground or deep water. Organisms may not be evenly spread, so it's hard to choose good sampling spots. Some species are hidden underground or well camouflaged, which makes them easy to miss. This problem can cause mistake in counting, which affect the accuracy of the data collected. After that is question number 5. Why is there a difference in the number of plant species in the quadrat? The number of plant species can be different in each quadrat because of sunlight. Some areas get more light, helping certain plants grow better. After that is soil. Different soil types or moisture level affect what plants can survive. After that is competition. Big or fast growing plants can block others from growing. For the number 4 is a human activity. People stepping on or damaging plants can reduce species. After that is microhabitats. Small change in land like slopes and wet spots affect the growth. After that is seed spread. Seeds may fall unevenly due to wind, animals or gravity. So both the environment and living thing affect how many species grow in each quadrat.